Good morning all. So today we are going to discuss mechanism of antibiotic resistance in bacteria. So antibiotics are critical tools for preventing and treating infections which are caused by specific bacteria in people, animals and crops. In healthcare, antibiotics are one of the most powerful drugs for fighting against life-threatening bacterial infections. Now, what is antibiotic resistance? Antibiotic resistance happens when the germs no longer respond to antibiotics, which are designed to kill them. That means the germs are not killed and continue to grow. It does not mean our body is resistant to antibiotics. The bacteria and fungi are constantly finding new ways to avoid the effects of antibiotics used to treat infections they cause. Infections caused by antibiotic-resistant germs are difficult and they are sometimes impossible to treat. In many cases, antibiotic-resistant infections required extended hospital stays, additional follow-up doctor visits, and costly and toxic alternatives. Now, how can taking antibiotics contribute to antibiotic resistance? Anytime antibiotics are used, they can contribute to antibiotic resistance. This is because increases in antibiotic resistance, those are driven by a combination of germs which are exposed to antibiotics and spread of those germs and their mechanism of resistance. When antibiotics are needed, the benefits usually outweigh the risk of antibiotic resistance. However, many antibiotics are used unnecessarily and misused too, which threatens the usefulness of these important drugs. For example, too many antibiotics are being prescribed unnecessarily to humans in US. CDC estimates about 47 million antibiotic courses are prescribed for infections that don't need antibiotics like cold, flu, etc. That's about 30% of all antibiotics prescribed in these settings. Everyone has a role to play in improving antibiotic use, and appropriate antibiotic use helps to fight antibiotic resistance and ensures that these life-saving drugs will be available for future generations. Now, why should I care about antibiotic resistance? This antibiotic resistance can affect many persons at any stage of life. People receiving health care or those with weakened immune system, they are often at high risk of getting an infection. Now, antibiotics antibiotic resistance, they may jeopardize the advancement in modern healthcare that we have come to rely on, such as joint replacement, organ transplant, cancer therapy. These procedures have a significant risk of infection, and patient won't be able to receive them if effective antibiotics are not available. So, antibiotic resistance also impact not only the medicinal field, It also impacts veterinary and agriculture industries. Now, how can I protect myself and my family from antibiotic resistance? Protect yourself and your family from antibiotic resistance by doing your best to stay healthy and keep others healthy too. Then cleaning hands, covering coughs, staying home when sick, and getting recommended vaccines such as flu vaccines. Taking antibiotics only when they are needed. That is an important way you can protect yourself and your family from antibiotic resistance. Talk to your doctor about the best treatment if you are sick. Never pressure your doctor to prescribe an antibiotic. When antibiotics aren't needed, they won't help you and their side effects could still cause harm. So you should ask your doctor or pharmacist about the steps you can take to feel better when an antibiotic isn't needed. If your doctor decides an antibiotic is the best treatment when you are sick, take the antibiotic exactly as your doctor tells you. Do not share your antibiotics with others. Do not save them for later. Talk to your pharmacist about safely discarding leftover medicines. And do not take antibiotics prescribed for someone else. This may delay the best treatment for you and make you even sicker or cause side effects. And you should talk with your doctor and pharmacist if you have any question about your antibiotics. Now, coming to the mechanism of drug resistance. The bacterial resistance. It is the capability of the bacterial cell to prevent antibiotic, bacteriostatic or bactericidal effects caused to the bacteria. The four main principal forms of antibiotic resistance are natural resistance, acquired resistance, cross resistance, and last, multi-drug and other types of resistance. First of all, about natural resistance, that is intrinsic or structural resistance. In this type of resistance, the usage of antibiotic, it is not associated with resistance. This can be caused by the bacteria's structural property. Some bacteria are intrinsically or they are naturally resistant at the time of the formation itself. So, this type of resistance due to the bacterial structural properties that may result in intrinsic resistance or microorganism which doesn't follow the target antibiotic structure or the antibiotics which due to its characteristics do not encounter its target. Then gram-negative bacteria and vancomycin. These are the two examples showing antibiotic showing natural resistance. In this, vancomycin antibiotics are actually given to gram-positive bacteria. They does not move through the outer membrane, so that these gram-negative bacteria are naturally insusceptible to vancomycin. Likewise, L-form bacteria, that are cell wall types of bacteria, cell wall less type, because they doesn't possess any cell wall. And also ureaplasma, mycoplasma, these forms are also natural cell wall less types. And this mycoplasma that are naturally owing beta-lactam antibiotics. So this mycoplasmas, because of the lack of the cell wall, they are not susceptible to beta-lactam antibiotics. So they are exhibiting beta-lactam antibiotic resistance. Next is acquired resistance. Regardless of the resistance development, so the acquired resistance, it is mainly due to the alteration in the genetic features of bacteria. That means the bacteria acquire certain properties and acquired because it is not affected by the antibiotics. It was previously susceptible to it. So after acquiring this resistance, bacteria now becomes resistant to the particular antibiotic in which it was susceptible when they are taken previously. So this form of resistance comes from main chromosome or extra chromosomal structures, that is either from plasmids or transposons. The chromosomal resistance results from the if this main form of resistance is from chromosome, this chromosomal resistance, it may result from either from mutation that can change this randomly the bacterial chromosome, and these mutations can occur by certain physical and chemical factors. And these mutations occur by physical and chemical factors, and this may be due to the changes in the composition of bacterial cells. So these are the several reasons. One can be either through main chromosome or extra chromosomal structures. Then another is change in the composition of bacterial cell. So that may be decreased bacterial drug permeability or maybe changes due to the drug's target in the cell. Example for this type of acquired resistance is seen in streptomycin and aminoglycosides. Erythromycin and lingomycin can develop this resistance, this type of resistance. So this type of resistance are shown by those antibiotics. Now extra chromosomal resistance. So we have discussed chromosomal type of resistance, acquired resistance. Now what will be the extra chromosomal resistance? That may rely upon extra chromosomal genetic materials that can be transmitted through plasmids, transposons and integrons. These are the three types of extra chromosomal materials which are responsible for causing extra chromosomal resistance. First one is plasmid. Plasmids are mainly the segments of DNA that can replicate independently of chromosomal DNA. So within a DNA, there can be a chromosomal DNA and plasmids. These plasmids can replicate independently, and it is typically responsible for developing antibiotic inactive enzymes. Plasmids, because they acquire certain properties. There are main forms of holding genetic material. Actually, these plasmids hold certain materials, genetic material for drug resistance and etc. And these are the main forms of holding genetic material from the bacterial cells. And this form are transduction, transformation, conjugation or mechanism of transposition. അതായത് ഒരു ബാക്ടീരിയയിൽ നിന്നും വേറൊരു ബാക്ടീരിയയിലേക്ക് ഈ ഡ്രഗ് റെസിസ്റ്റൻസ് പ്ലാസ്മിഡ് എങ്ങനെയാണോ ട്രാൻസ്ഫർ ചെയ്യുന്നതെന്ന് വെച്ചാൽ ഇറ്റ് ക്യാൻ ബി ഐദർ ത്രൂ ട്രാൻസ്ഡക്ഷൻ പ്രോസസ്, ട്രാൻസ്ഫോർമേഷൻ, conjugation and mechanism of transposition. The genes with antibiotic resistance on the chromosome or plasmid are intertwined and are situated at the beginning with different integration groups. So next group is about integrons. And in this integrons, recombination is very normal in integrons. സോ അപ്പോൾ ഈ എക്സ്ട്രാ ക്രോമോസോമിലും അതേപോലെ തന്നെ ക്രോമോസോമൽ റെസിസ്റ്റൻസിന് പുറമെ integrons are also taking part in acquired resistance. and recombination is very normal in integrons. So that's all about